Good morning students. Today we are going to see about the plant mediated synthesis of nanoparticles. So we are not known that what is nanoparticle. Nanoparticle comes under the range of 1 to 100 nanometer. So we have synthesis the nanomaterials in two approaches. One is bottom up approaches and then another one is top down approaches. In bottom-up approaches, we have to arrange the smaller components into more complex assemblies. So the bottom-up approaches examples are chemical and physical forces operating at the nanoscale to assemble the basic units into the large structure. So we are using bottom-up approaches, we have to synthesize the carbon nanotubes. In top-down approaches, we have to create the smaller devices by using larger one to direct the assembly. The most top-down approaches to fabricate involves the lithographic patterning technique using short wavelength for optical sources. So top-down approaches is the main example for biological synthesis of nanoparticles. So here the pictorial representation of top-down and bottom-up approaches. So top-down approaches, the bulk material change into the small nanomaterials. In bottom-up approaches, the small atoms we are converted into the nanoparticles. So material what we will take, that is the main things in the nanomaterial synthesis. So we are using the top-down and bottom-up approaches which comes under the range of 1 to 100 nanomaterial. We are called as that particle is a nanoparticles. So for nanoparticle synthesis, we are using three methods for synthesis the nanomaterials. One is physical method, another one is a chemical, the last method is biological. The plant mediated synthesis of nanoparticles comes under the biological method, which having the many advantages for that biological method. Because in the physical and chemical methods, we are using some of the chemical reagent for synthesis of nanoparticle, it having the side effects. In biological methods, it is very useful. It is very compatible to the human system. That's why we are all of you mostly using the biological methods of nanoparticle synthesis. So our topic is plant mediated nanoparticle synthesis. Here where you how the plant material prepare for that nanoparticle synthesis. So here for example I am taking Aegil marvelous leaf extract for the nanoparticle synthesis. So first the Aegil marble, 20 gram of Aegil marble leaf extract leaves were taken and washed, air dried and chopped into the fine pieces. Then finally cut the leaves or dissolved in the 100 ml of distilled water. After it will be taken in the after we will place in the water bath for 45 minutes. Then after collecting that extract, I am using the filtrate. Then obtained filtrate I am using for the nanoparticle synthesis. The nanoparticle synthesis, here the major components are present in the leaf extract of the Aegil marvelous, because it is the act as a reducing agent for nanoparticle synthesis. So Aegil marvelous leaf extract contain many secondary metabolites such as alkaloids, phenolics, flavonoids, sugars, saponins. So before we are going to synthesize the nanoparticle, we have to check the secondary metabolite analysis in the leaf extract. So for alkyd extraction, we are using the confirmatory test of Wagner's test. It will be changed the brown color precipitates. Then phenolic compound test analysis, we are using two methods, ferric chloride test and the lead acetate test. For ferric chloride test, it will be changed into the deep blue color formation. For lead acetate test, white precipitate will be observed. For flavonoid test analysis, the result observation yellow color changes in the slide. It will be shows the presence of the flavonoids. For sugar analysis, we are using filling reagents for confirmation of the particular sugar present in the leaf extract. For saponin, we are performing the foam test. Present of the foam indicate the saponins are present in the leaf extract. This is the some color changes observation for phytochemical analysis observed in the leaf extract of the Aegil marvelous. So most of the phytochemicals, flavonoids, alkaloids, saponins are present. Here we are taking that plant leaf extract for the nanoparticle synthesis. So these are the pictorial representation concentration of how do we are synthesis that particular nanoparticle by using leaf extract of the Aegil marvelous extract. So 10 ml of the leaf extract we are taken, then 90 ml of silver nano solution, silver nitrate solution were taken. Then after we have to mix, immediate color change was observed. After addition of the two solution, the resulting mixture is incubated at room temperature for 24 hours in a dark room for precipitation. Then after reaction is complete, the precipitate is centrifuged 5000 rpm for 15 minutes. The supernatant is discarded and then pellet were washed with 70% ethanol. Then after we have to air dry the pellet and then we are used that is a silver nanoparticles. So this is the color change observation. The first one is brown is indicate the Aegil marvelous leaf extract. The white color, milky white color represent the silver nitrate solution. Then after mixing of 3 hours, it will be changed to yellow color. After 24 hours of incubation, it will be complete precipitation are observed. This is a effect of which contain the fine particle of the nanoparticle after completion of the air drying of the particular nanoparticles. So before the synthesis, we have to optimize the various parameters for synthesis that particular nanoparticles. For optimization, we are using the increase in the concentrations of silver nitrate and the temperature. After we have to addition of the two solution, we have to incubate 30 C and 60 degree C, 90 degree C and 120 degree C. Then we have to check the pH of that solution also 5, 7, 8, 9. Then we have to incubate the two solution after addition 6 hours, 12 hours, 8 hours and 24 hours. Then ratio of that plant extract and silver nitrate solution also monitor in the nanoparticle synthesis. So after optimization, we have to confirm. So which is the best suitable environment for producing the silver nanoparticle synthesis. So this is the optimum optimized parameters required for the silver nanoparticle synthesis by using Aegil marvelous leaf extract. So concentration of AgNO3 is 1 millimolar, temperature 60 degree and then pH 8, incubation time 24 hours and the ratio of plant extract and AgNO3 is 1:9. So this is the optimum parameters required for the silver nanoparticle synthesis by using plant leaf extract. Thank you.